My name is Christine Gebsky. I'm with Repligen Corporation, and today I'm pleased to be speaking about the impact of our technologies in both upstream and downstream processing, and looking at the process productivity and economic benefits of the diverse technologies that we offer. So by way of an agenda, I'm going to give you a brief introduction to Repligen Corporation, one of the newer bioprocessing tools providers in the market. We'll then look at, very simply, the upstream technologies and considerations, and then downstream technologies and considerations. So Repligen's been in the bioprocess space since the mid-1980s. Over the past 4 to 5 years, we've been retooling our organization to focus on being technology leader in providing innovative tools and services and technologies into the bioprocess space. From a legacy standpoint, we provide most of the world's recombinant protein A that's used to purify monoclonal antibodies, but we've become an industry leader in cell culture process intensification, pre-packed chromatography, column technology, and single-use tangential flow filtration. And very recently, we merged with Spectrum Labs, and are now the only bioprocess tools provider that provides both hollow fiber filters and flat sheet filters. So we have a very exciting future ahead of us as we begin to bring all of these technologies together. From a business standpoint, you can see where our focus lies. We have chromatography solutions, filtration solutions, and protein solutions. So entrenched in this space in the biopharma industry and bringing our tools from multiple global manufacturing sites. If you look at the legacy of the company, we've had rapid growth through both organic growth, launching innovative products, and executing on strategic acquisitions. Of note, acquisitions of Refine technology that brought in the Excel ATF assets, Atoll GmbH in Germany that brought in pre-packed column technology to add on to our existing Opus technology, and then most recently, as I mentioned, Spectrum Labs. So our products are used in clinical and commercial applications, and as I've noted, span upstream and downstream processing. That's depicted here. We have scalable technologies that really map to the bioprocess workflow that our customers, such as yourselves, utilize in upstream processing, downstream processing, and analytics. And we really can provide technologies to a diverse range of biomolecules that the industry is working with. So that was a bit of background on Repligen as a whole. If we think about providing tools that bring process productivity and economic benefits to the market, we want to think about some Lean Six Sigma manufacturing principles and understand how we can begin to optimize processes and reduce time spent prepping for bioprocessing and actually executing that work. So some of these important principles, one is the law of velocity. Here the goal is to reduce waste by reducing WIP. And WIP will lead to, the more WIP that you have in a process, the more downtime, wasted time, and lead time that you have in a process. And then looking at complexity, if you can keep processes simple, you reduce complexity, and that helps you improve your WIP and your waste. And really looking at the law of focus, which is a Pareto type principle, where you're looking at 20% of the activities that can cause 80% of the delays, and figuring out how you can optimize and streamline processes to improve productivity and cost of good economics. Okay, so if we transition to upstream technologies, I'll introduce Excel ATF. This is a cell retention device, filtration device that allows for high cell concentrations and high protein concentrations to be attained. The product's available in both single-use and stainless formats. It is the leading perfusion device in the marketplace and allows for significant improvements to the upstream processes from cell banking to true perfusion cell culture. If we look at the way the technology functions, we have a alternating tangential flow diaphragm pump affixed to a hollow fiber filter. This diaphragm pump moves the cell culture mass from the reactor into the lumens of the hollow fiber filter, and then a filtrate pump removes clarified permeate from the cell culture mass, and the cells are returned to the bioreactor. So ultimately, we can attain very high cell densities, higher productivity, longer cell culture runs, lower capital investments, ultimately lower operating cost, and ultimately derive better process economics. So multiple ways that this technology is applied in upstream cell culture. One of the first ideas is in cell banking. We can produce a high-density cell bag that can be used to eliminate additional seed train steps and actually inoculate a seed reactor at a higher cell density and allow for reduction of seed expansion steps, which leads to time savings and risk reduction in that operation. By removing multiple steps in the seed process, we can also reduce the amount of handling and manual processing and ultimately reduce risk in this type of process. You can then expand that technology to N-1 seed train optimization or N-2. Here, when you have your reactor just before the production reactor, you can use ATF cell retention technology to increase the cell density of that seed reactor and then seed your production reactor at a higher cell density, ultimately reducing the time it takes in the production reactor to get to the cell concentrations and product titers that you're interested in. And this technology can be applied to either fed batch cell culture or perfusion cell culture, which is important consideration for the way proteins and vectors are produced. So if we look at some process economics, if we're comparing a fed batch cell culture process and a perfusion cell culture process, you can see that there's differences in the scales to which you need to operate the seed train. The production bioreactors in a perfusion process are much reduced in terms of volume, which leads to capital equipment savings and ease of operation. Your product titers will change, but ultimately you're producing more protein more consistently over a longer period of time. So you can save in the seed train and then be producing in a more steady state fashion for longer periods of time. Ultimately, producing product for lower costs and in a smaller amount of space, as is depicted here. So here, comparing the benefits of perfusion technology as compared to fed batch, you can see the capital investment for both disposable reactors and stainless steel reactors. You can see the significant reduction in capital required and in operating space required if you're running perfusion technologies compared to fed batch technologies. Okay, so if we look at some performance features, again, this is perfusion versus fed batch cell culture. On the left, we're looking at viable cell density as a function of culture time, and on the right, the cumulative protein as a function of culture time. You can see that with the fed batch reactor in blue, you have the cell densities decay over time, and the cell densities are lower, and the viability decays over time, and you have lower protein produced. So ultimately, by day 10, the cell densities in the ATF reactor are 5-fold higher than the cell densities in the fed batch reactor, and we can produce 6 times the amount of protein, cumulative protein, with the perfusion process. So this all leads to productivity gains in upstream processing, and a way to consider producing proteins more efficiently. Okay, if I go back to the N-1 application, here the goal is to seed the production reactor with a higher cell density, and once the cell density is about 10 million cells per mil, we can then seed the production reactor, and ultimately we're able to get to higher cell densities with a high seed culture in a shorter amount of time. So if we seed with a low seed, it takes, we can only get to about 20 million cells per mil over 14 days, but seeding with a high seed culture, we can get to over 25 million cells in 7 days. So again, demonstrating the benefit of high seed, seeding at a high density into the production bioreactor. Okay, that high seed culture was able to produce the same amount of protein titer in 7 days as compared to 14 days that was required with the low seed titer, with no difference in the cell-specific productivity. So ultimately, seeding with a high density can produce more protein in a shorter amount of time, again driving a productivity gain into your processing. Okay, the benefits of single-use systems in the industry are well established. Again, going back to those lean principles of reducing WIP and allowing or implementing single-use technologies allows for reduction or elimination in the steps that are non-value add to the processing that the industry is looking to do. We now have a full range of XCell ATF single-use systems available, having recently launched the XCell ATF 10 single-use system, which you can come by our booth and look at. This technology, the single-use technology, mirrors our stainless steel technology. We have 2 hollow fiber filter types available, 0.2 micron PES and 0.2 micron PS. And ultimately, as I'll demonstrate, implementing single use allows for a significant reduction in the implementation time associated with getting this cell retention device configured into your cell culture operations. Scalable from lab scale to production scale, and the single use technology mirrors the stainless steel technology in terms of fit, form, and function. So if we look at the process that's typically required to implement stainless steel XCell ATF in a manufacturing operation, multiple steps associated with prepping the device, cleaning, autoclaving, integrity testing, and when you look to implement single use, you can see that many of these steps are eliminated from the process, and therefore you can derive over 80% savings in setup and decommissioning time, and over 7-fold savings in the labor hours that are required to implement the technology. So deriving a polycarbonate-based single-use system for XCell ATF will help our customers reduce the WIP operations that they have in their suites and derive lower support times to implement this technology. Okay, so transitioning to downstream, I wanted to speak about the range of pre-packed columns that we offer. So the Opus product line is offered from 50 microliter packed beds to 85 liter packed beds, going from half centimeter diameter columns to 60 centimeter diameter columns. This is a open platform user specified, therefore customizable product line. It's the broadest range of pre-packed columns in the marketplace today, and customers are able to choose your diameter, your bed height, and the resin type that you would like to work with. So it's a really platformable pre-packed column solution that, as I'll demonstrate, is driving productivity and economic benefit into downstream processing. Okay, so we've been working with pre-packed column technology since about 2012, having developed the capability to pack or up to 60 centimeter diameter columns. And from our production statistics here, you can see that we are leaders in this technology, and at this point have packed many, over 800 5 to 60 centimeter diameter columns provided globally to the bioprocess industry for the purification of many biologics. On the small scale, Opus PD development scale of pre-packed columns, we've packed tens of thousands of columns that are utilized for process development around the globe. The products are used in commercial processes and over 100 phase 1 through phase 3 campaigns, and ultimately we work with over 100 different resins for the large scale columns and over 300 different types of resins for the process development scale columns. So the benefit here is, as I'll walk you through, is driving optimizing process economics, reducing costs compared to self-pack columns, and bringing time savings into both development settings and GMP manufacturing settings. Okay, the Opus PD range, as I've noted, is shown here below, from 50 microliter robo columns for high throughput parallel process development and sample prep, to Valleychrome columns, 3 different diameters from half centimeter diameter to 1.1 centimeter diameter, used for scale up and scale down validation. So if we look at the benefit of RoboColumn technology in process development and sample prep, this is some recent data published between Coherus and Tecan, which you can find on the Tecan website. We partner with Tecan in their high throughput liquid handling technology, and the marriage of this liquid handling workstation and Opus RoboColumns provides a elegant solution to improving the time required for process development, parameter screening, and sample prep in different workflows. So this customer had implemented these technologies and found an 8-fold increase in throughput compared to manual methods for their sample prep and process development activities. And previously, Amgen has published on the use of RoboColumn and Tecan technology, looking at that combination of tools compared to ACTA systems with standalone columns, and has shown the productivity gains in terms of the number of samples that can be processed on a time basis and the cost savings per column. So this is a high throughput technology that really drives productivity gains into parameter definition for downstream processing and sample prep applications. If we scale up slightly to Opus Valleychrome columns, these columns can be utilized for scale up and scale down activities. Multiple applications here, most associated with phase 1 and phase 3 type validation activities, from scale down modeling to resin lifetime assessment to viral clearance studies. And if you model the column packing activities required to support a single viral clearance study, you can see potentially 3 chromatography steps that would be studied, needing 3 columns per per virus or per arm of the viral clearance study. You may need a backup column so that you're not wasting time in the suite when you go to execute your work. If you're evaluating 4 viruses per chromatography step, an end user may need up to 36 columns to execute 1 viral clearance study. If you think about the number of hours to pack each column in a process development setting, you now have a highly trained process development scientist or engineer spending 164 hours packing and qualifying columns to support a single viral clearance study. These activities also do not include activities such as hardware acquisition, column cleaning, documentation. That's all required to support that work. So with the use of Opus ValleyChrome columns, you can decouple all of that work from your workflow. We can provide a finished good column to your specifications with the resins that you need and ultimately drive productivity gains into your operations in support of viral clearance studies. Okay, we have an extensive model that we've developed with BPTC to look at the cost and labor and suite considerations for implementing large-scale columns. This is a proprietary model that Repligen has built, but we work with customers to individually to assess how pre-packed column technology can impact the work that you're looking to do in clinical manufacturing. So if you're thinking about implementing pre-packed columns, it's not just the act of packing and qualifying a column, but there's many supporting steps that go along with column packing listed here, right? So some of them you need IQ/OQ documentation around the hardware, around the actual packing and testing of columns. You may be you're supporting resin preparation, buffer preparation, and then if you're looking to reuse columns for other purposes, you have all the workflow around unpacking, column cleaning, and that column cleaning validation. So many steps that support the column packing operation that sometimes are missed when considering the use of pre-packed versus self-packed columns. So, but the real cost of column packing is sometimes more than a mathematical model can capture. So at the end of the day, column packing is not a value-add activity. It's a WIP activity when you're thinking about lean manufacturing, when you're looking to produce a biological molecule. And aspects that the model does not capture include packing issues, right? If you're having to repack a column multiple times to get that column prepared for your clinical or commercial manufacturing, that can be a huge draw on an organization. The opportunity cost, right? So you're using staff and facility for self-packing activities, which can be considered non-value-add activities. You're also keeping maintenance records on equipment, having to purchase capital equipment and sign-off procedures associated with that. So there's significant effort if you think about what it takes to go from I need a piece of equipment to pack a column to having a qualified column prepared for GMP manufacturing. So if we look at our model, I have 2 considerations this morning. The first is using our 45 centimeter Opus column when you're comparing to a self-pack that would include or utilize a pack-and-place type column, such as a ChromaFlow column, and importantly, where the column is going to be dedicated to a defined process or product, right? So where you're not looking to reuse that and therefore clean and validate that cleaning of the column for other processes. So from the model, which has many components, including suite time, labor time, materials, the resin cost, the column hardware cost. You can see that using Opus pre-packed column technology drives about $85,000 worth of cost savings, over 50% per molecule, as compared to self-packed, and over 135 FTE hours when compared to self-pack processes. And ultimately saving over 2.5 days of sweet time. So eliminating the non-value-add WIP type activities associated with column packing and implementing a finished good OPUS column can bring significant cost and labor savings into your facility. And ultimately with the sweet time, you can be doing more bioprocessing versus non-value-add WIP activities. If we then look at that same model, so 45 centimeter diameter columns, pack in place, self-pack technology compared to OPUS, but here the columns will be utilized for multiple molecules. So in this part of the model, 3 projects per year. That now means you're adding in the activities associated with column cleaning and cleaning validation, so that changeover to use the hardware for another molecule. But even in this case, you can still see we're driving, enabling cost savings on a molecule basis and the same amount of FTE hour savings, again, removing those non-value added activities. And ultimately, almost 3 days of sweet time being saved and therefore put back into the pool of time that can be used for bioprocessing. So ultimately, OPUS prepacked columns can bring significant cost and sweet time and labor time savings into your facilities. Okay, if we move further downstream to tangential flow operations, typically used for protein, biomolecule concentration and diafiltration, our Tangenics line of CS and Pro TFF cassettes. CS cassettes are single use and Pro cassettes are reusable. So I'll focus on the benefits of the single use technology. This technology came to us from Tangenics. They've been serving the marketplace since 2007. Products are used in hundreds of clinical processes, 5 commercial processes, scalable formats from 0.1 meters squared to 2.5 meters squared, and 2 membrane chemistries. And ultimately, with the single-use technology, we can again derive significant process economic benefits compared to reusable flat sheets. Okay, so one thing I didn't mention is that the cassettes are compatible with the hardware that exists from other filtration providers, and ultimately these are the first purpose-built single-use TFF cassettes for tangential flow filtration. So what that means is that because of the mode of construction, we can offer these cassettes at an 80% reduction in cost compared to reusable cassettes that are on the marketplace today from different suppliers. So about a 5-fold decrease in cassette cost on a per meter squared basis compared to reusable cassettes. So the economic benefit to that is obvious. Okay, with implementing single-use TFF technology, again, we can reduce the waste in the WIP in terms of the steps that are required to move from setup of TFF cassettes to storage of TFF cassettes. So here you can see with single-use technology, we're reducing or eliminating, many of the supporting steps around sanitization, flushing, checking the normalized water permeability, cleaning, so we can have over a 50% reduction in the labor required to execute these processes and 40% reduction in the time required to work with these products and run them. And importantly, you can then execute a TFF operation in a single shift, so about less than 8 hours compared to longer shifts. So ultimately, looking at diverse and enabling technologies across upstream bioprocessing and downstream bioprocessing, highlighted the economic and productivity benefits of our Excel ATF technology, from cell banking to seed train to perfusion cell culture, using this cell retention device to derive productivity gains and economic gains in upstream. On the downstream side, we've looked at OPUS, both process development and large-scale pre-packed column technology, and demonstrated how these tools can be used for high-throughput process development and sample prep, bringing 8-fold increases to the throughput of those of that process development. And then looking at the significant labor, time, and suite savings that the implementation of large-scale pre-packed columns compared to self-packed columns can bring to bioprocessing. And then closing with the benefits of single-use TangenX TFF filters in reducing costs, labor, and buffer usage in TFF operations. So with that, I thank you for, again, your dedication and enthusiasm at this point in time.